again, I'd like to thank you, and Angie, for agreeing to do this webinar on the cognitive paradigm ontology. Now, as you can see, and I would like to thank everyone that is attending today. The Mind Research Network and UT Health Science Center at San Antonio are both places where there are lots of very interesting, especially to cognition, projects going on. These are both places which take data from imaging of actual humans. And one of the biggest problems with some of this data is that everyone has their own sort of cottage industry kind of a way of describing these things. So it is actually a very difficult thing to try and put some of this data together. So with that in mind, I think I'll do a long introduction of either one of you, as most of you are very well known. So please take it away.
Yeah, we're going to be trading back and forth. I have a couple things to say, then Angie will present a little bit more detail about certain aspects of this project, and then I'll come back and wrap it up. So first, one of the key issues for us in cognitive neuroimaging is finding out how to share data. I mean, that's one of the driving forces here. And the human imaging database and the XNAP databases that store a lot of imaging data, and we have had dramatic issues trying to get data from both databases. Let's say we want the anatomical scans or the T1 weighted scans. Usually we end up building a data model. You know, how do we access, how do we find the T1 scans in each database, and that allows us to solve that particular problem. But of course, you say, well, I actually want to know, this is the kind of problem that the Neuroscience Information Framework wants to deal with. We want to find any MRI data that might be in databases, and then we need to know, okay, well, some of the T1 weighted, T2 weighted, MEG data, EEG data, MRS data, DTI, SCID information, what if these are actually MRI related data? And that, of course, leads to wanting to know the semantic information and the link to ontologies. And then getting beyond that, of course, when we get, let's say we actually retrieve some of the analyzed results that we might be interested in, and okay, well, we have some subject information, demographic information, clinical assessments, what cognitive tasks were they actually doing in order to get this kind of fMRI signal out? How was the MRI data collected? How was it analyzed? And where are we actually seeing the statistical results that we might want to integrate across different experiments? Now, obviously, this gives us a lot of reasons to build taxonomies, hierarchies, ontologies, etc. And the area that Angie and I have been focusing on in particular is representing details of the cognitive paradigm. And of course, beyond actual data retrieval and understanding what we would like to understand, really, what are we looking at in the data and the results? How does that lead to new hypotheses in the field? How can I actually not just retrieve data, but actually integrate the information that comes with that data? And of course, so there are a lot of ontologies. We've been talking about a few in this series. OB covers experimental methods, RADLEX covers imaging methods, CogPo, which is mine, cover cognitive experiments, NEMO, that Gwen represented, is already doing EEG and MEG, and there's others for other parts parts of the issues that might be needed to really understand what's going on. And of course, the NCBO bioportal can be used to explore these and other examples, and NIF is in there as well, covering a number of things. Particularly in modeling neuroimaging data, has basic categories for most. What is a diagnosis? What is a human subject? What is a T that might be an imaging parameter? What are the characteristics of a neuron or a cell that we might care about? CogPo gets into more details of the experimental paradigms. What are we talking about when we say experimental condition? What is a response? Cognitive Atlas, that Russ will hopefully be talking about next week, includes terms for cognitive processes like working memory by their definitions. And of course, community input is absolutely necessary, as we all know, for coming up with agreements on the we want to be able to either search or reason for about correctly. It expands the future without introducing errors, which has been a really interesting question. And of course, there's a question of granularity in modeling our data. Do we need to represent the difference between a visual checkerboard that flashes regularly at 2 Hertz and irregularly at 1.5 Hertz? No, the DTI scans have different direction sets. Does that actually affect what we want to reason over? Do we need to represent exactly how increasing or decreasing a TR value is going to affect the MR scan? Certain people need that, certain people do we need to be able to take into account the differences in diagnoses that exist between these different diagnostic standards, and all of the hairy details between neural metabolism, and it really depends on what your goal is in modeling this neuroimaging data for data sharing and for reasoning. And of course, goal number 1, and in fact, which Angie will talk about more, is meta-analysis, retrieving the right results and analyzing them together to identify really what is the robust finding in the field. The next goal after that, oops, I'm sorry. I'll try to tell me if you miss something. When I cut out, of course, I.
Yeah, it's you are cutting out just a little tiny bit. We're trying to get a little better, but maybe just stepping away a little bit from the computer might help.
Unfortunately, I'm wearing a headset.
Oh. Well,
so the microphone is sort of fixed where it is.
Okay. All right, that sounds good.
I can try to sit up farther ahead from my head, but that's going to be tricky.
I can see that. Okay. Yeah.
Oh, maybe turn down the volume. I could do that. I could do that.
Just turn down the volume just a tad, and then maybe.
Does that help?
Oh yeah.
Or is that too weak now?
No, that's much better.
Okay. Good. Well, I'm sorry you guys didn't tell me beforehand. I could have turned it down a few slides ago. But anyway, the interest that I have primarily is mega-analysis, retrieving the right imaging data sets, along with what I need to understand what it is that they did, so that I can do analyses. And it sounds pretty trivial, but it's not really solved yet. There's a lot of folks who need to be working on this to identify, for example, what all do we need to know about structural imaging? What do we need to know? What's a data model for spectroscopy? And where do they coordinate with each other? And we're trying to do that as part of a number of different data sharing efforts. And goal number 2 is retrieving and reasoning over results from various analyses. So if we have several experiments, for example, show activation in a brain region like Brodmann area 7, and we know the details of another experiment, what can we actually predict results? Can we identify what we would, can we to saying, you know, we should see activation here, here, and here. We should see a diagnostic effect or something like that. And the current standard on how to do that involves many, many human hours of extracting the information, curating, and annotating it within a very carefully constructed centralized knowledge repository. Which obviously needs to be built upon, because in order to really reason about this, we're going to need to move beyond single knowledge bases, and we're going to need some interoperability on what the relationships are between different concepts and ways of thinking about things. Now, Angie, is this yours? Is this where you wanted to start? Okay, I'm going to hand this over to Angie then. So I should stop sharing, and you can share.
Yeah, this is it. Can we take over?
Yeah.
We good? All right, so taking the reins over from Jeff, this slide is intended to really sort of focus on specifically what CogPro's role is in our community. For us, it was really important to think about the overall goal of the community, which is to determine structure relationships, understand which brain regions are responsible for which cognitive goals. Game being potentially a deliverable of a probabilistic functional atlas that really maps out these relationships in a very quantitative way. But it's not that simple in the sense of this is a many-to-many mappings problem, in that a single region can be involved in many different cognitive processes, and a single cognitive process usually activates multiple regions, networks of brain are multifaceted in the types of functions that they're responsible for. And so to do this, we need, for our field, we need database synthesize all the data we've been collecting. We are collecting massive amounts of database information on in order for these databases, we need to do ontology. So if we're going to look at a specific type of query that can be posed across the different databases, take for example, find the data examining the relationship between hippocampal volume and Alzheimer's disease. And currently, we have three main components of this search. We need to know what hippocampus is, what volume actually means, what Alzheimer's disease means. And there are current ontological projects on to address neuroanatomy, imaging acquisition, clinical diagnosis. These processes, these sorts of representations are being addressed. Yes?
Angie, can I stop you for a second? I think, Jeff, can you turn off your microphone for just a second? Because I think we're getting a little feedback. So.
Okay. Yes, are you off?
Okay, I guess let's try again.
Okay. And so for this particular query, we have processes being made. But when cognitive was brought into existence, we were worried about queries such as find the data the neural activations were observed during sustained attention tasks. And right now, the whole idea of a representation for something like sustained that was a distinct omission in our graphical development. And so the cognitive paradigm ontology was born. It aimed to represent only the past being humanized and functional neurons, along with a meaningful decomposition of how we can talk about those tasks without enabling specific field-specific jargon. Like, there's the street task and the NBAC task and the Sternberg task, and each of these tasks have names, but we really wanted to get at the idea of
Angie, do you want to hold up for a minute? For some reason, you're getting a lot of feedback, or not feedback, but background noise. Did your microphone come a little unplugged or anything?
Have you had yours off, Seth? Anita, what do you suggest?
Anita, did you turn everybody down, or did I just turn myself down when we were trying to turn things down earlier?
And I was okay before?
Yeah, I can't hear Anita either, unfortunately. But you're very soft. You're very soft, but there's a hum in the back. Yes, you were fine in the beginning, but it's almost like there's static on the line.
Angie, can you just try to mute and then unmute again? You are in two separate states, so. Because I'm wondering if there's just feedback. Okay, yeah, I think there was some sort of a feedback going through your system. So go ahead and unmute yourself, maybe.
How about that?
That's no good.
Okay.
Okay, how about that?
That's much better. Okay, let's try getting back to this. Okay.
Okay, my speakers are muted, so I won't be able to hear you guys. Okay. That's
It's really kind of hard to hear. Let's see. Angie, unfortunately, it's really hard to hear you. The rush. Do you want me to try to take it? Okay. Let me share my screen. And oops, hang on, I still have to share Microsoft. Give me a second. And from current slide. I think Angie had gotten to this point about these different ontologies that are involved, and we're more of a structural question. What we're, of course, within the functional imaging community, what we're doing is things like the data that examines neural activations during sustained attention tasks, which is where the cognitive processes issues come in. Now, with the cognitive paradigm ontology, we started with the BrainMap schema that we've been working on for a decade now, and that was started before, in which an experimental paradigm is really broken up into the conditions that make it up. So, for example, CogPO is the result of a collaboration between the BrainMap project and the original functional imaging Burn project that really dealt with the issue of fMRI tasks and how do you describe them. So we've decomposed our tasks into conditions, and each condition has a stimulus, a response, and a set of instructions. And then CogPO is designed to facilitate linking data, could be neuroimaging, as Gwen pointed out, could be EEG or MEG, data from similar cognitive paradigms across different sources. And one of the for example, we wanted to use was the one shown here, where we have the same behavioral experience, if you will, where subjects are shown a number of stimuli, and then there's a delay, and then they are asked to, they're shown another stimuli and asked to say, is that one of the items you had to remember? And they respond, no. So the overall flow experiment is very similar. In each case, you have conditions of encoding, delay, and retrieval, and there might be certain stimuli with a particular modality. It's shown visually, a particular type, such as letters. It might be digits. The response, then either no response or you have to push a button, an fMRI, to say yes or no. And of course, you're doing different things during these conditions. Here, you're at the items and trying to remember them for later. During the delay, your instructions are to look at this. Usually people are rehearsing what they just saw, but they aren't instructed to, so sometimes they aren't. And then, of course, here, their instructions are to either recall the item and determine whether or not it is one you've seen before. But the same experiment goes under several different names. In the literature, what CogPO is trying to do is to facilitate identifying when you've actually got the same task or you're trying to do the same thing. Now, the BrainMap database is really a database intended to deal with results, as opposed to other databases which are really dealing with the data. BrainMap, the BrainMap database pulls from the literature and archives the MRI usually or PET data about where the results are, what are the coordinates of the peak activation, and the metadata that lets us know where those coordinates came from experimentally. There's a number of different applications. I highly recommend you check out the website. A number of different applications to work with BrainMap. Sleuth allows you to actually search the database. Ginger Ale is the meta-analysis software. We'll say a little bit more about that. That's really, Angie's really driven that. A fantastic contribution to the research community. And Scribe is the software entering information and metadata about the papers that we can then search on later. And Angie, to pop up a message or something if I've forgotten to say something. Now, we do have a number of community standards on the normalization of how the brains are, and the locations tend to be reported in the paper. A standard methodology is the Taylor-Ack and Turneau coordinates from their atlas. And so many, in fact, what we are leveraging with the BrainMap base is the fact that results are reported in usually a tabular form in a standard coordinates, which makes it a little bit easier to understand what's going on. The other standard space that's commonly used is the Montreal Neuroimaging Initiative, MNI space. and there's well-known transfers between the and M&I, so either one is fine, and the BrainMap software actually takes advantage of that and will work either space as needed. And of course, so from the various papers, you can identify where things are, what their coordinates are, what the regions are, and how these things were created. Yeah, that's true. And due to growth, there's lots and lots and lots of these. And what they mean in the sense that sometimes it's just contrast, sometimes it's correlation. You need to be clear on exactly how they were done. And this is where we actually see some of the examples. Angie, I don't know if you can type in what this is actually in a particular analysis. Is she typing it in? We can use the, there we go. Let me just go back over here for a minute. Yeah, that's true. Oh, this is a word generation task. Okay, so they can actually extract from all of the papers dealing with a particular task where the areas of activation were reported, and you can see there's quite a few. Now, Scribe is using papers. The BrainMap database can. Oh, I thought it was more papers than that at this point, but lots of experiments and lots of locational coordinates. Sleuth allows you to search the database using the standardized terminologies, and then GingerRail provides the coordinate-based activation likelihood estimate for meta-analyses. Metadata the BrainMap stores, and this was really the backbone of what we were doing in CogPo, involves, of course, information about the paper, some free text about exactly what went on in there, and key and pieces of information about the groups of subjects. What diagnosis of some subjects? Is this a paper about schizophrenia, for example? Healthy controls, and then the actual conditions involved in the experiment, and a key about what a paper is. BrainMap terminology. The paper, the paper involves a number of experiments, and what are the conditions that are involved in all of the experiments is actually sort of separated. A lot of folks, yes, that's true. This did develop as needed to describe the actual data and results. And experiments are usually a contrast of conditions. So you list for the paper all of the conditions, and then in a particular experiment in which you have results, you can talk about what those conditions were in the results. Because, and this is for neuroimaging data, and in fact it's true of most experiments, you need to be comparing things. Does the condition map to the disease? No, not at all. Condition, experimental condition. So if I was looking at that working example for back here, oops, there we go. Where we're talking about the conditions, we're saying, okay, you're in the encoding condition, you're in the delay, you're in the retrieval. Diagnosis, not at the moment. It's been the diagnosis options have been developed on what is in what's been needed, but it could easily be mapped to specific ontological terms, like gender, handedness, language, etc. Okay, these are the general breakdowns. What is the name of the actual experiment? Which subjects are involved? Because sometimes in a healthy controls and several other groups. And you need to know which two you're really talking about when you find a particular spot, if you will, on the brain. What conditions are you contrasting? What was the imaging modality used? What was the actual contrast? For example, if we had encode versus recall, are we looking at encode greater than recall or recall greater than encode? It's one of the things that we do a lot of. Is there a paradigm class that we can actually map this to? What is the behavioral domain? Is there a context, such as this was pre-treatment or post-treatment or something like that? And of course, conditions is really what we've been focusing on as a commonality and a way to group the actual experiments. This is somewhat of a fixed set of menus. However, they can be added to if we need to. These are the stimulus modalities that it recognizes, the available instructions that are commonly used, the stimulus type that have been needed in order to represent papers that it uses, that it includes in its database, the response modality. Response modality is really the part of the body that was used in making the response, and the response type. What were you being told to do? Which is different from the instructions, right? Because you could be told, that you are going to imagine something, and then you might have no response. Or you could be told you need to attend to something, but your response is actually a button press when you see the thing you're looking for, and you might be doing it with your hand when it's a button press, but they're sometimes done with the foot. And yes, we do always mean response in the response and response modality. And yeah, we'll come back to the instructions in a little bit. Now we have a set of paradigm classes. These are the ones that have been built up, if you will, over time. And from the point of view of an ontology, it does lead to some interesting questions, but we'll come back to that. And we also have the behavioral domain. These are basically the fixed set of behavioral domains that are currently coded into the database scheme. It gives us the beginning of what are we actually looking at in this particular experiment. Are we looking at imagination? Are we looking at sex? Are we looking at disgust or something like that? Looking at the effects of alcohol or auditory processing, and it's an interesting combination. We know this is not a complete representation. Pharmacological is now independent. I'm glad to hear that. It always bothered me that it was a behavioral domain. But this is obviously the backbone. Okay, how's that? Are you back now? We good? Can you hear me? Excellent. Okay, thank you. Good. So, okay, I'm sorry. What I was trying to say was these is obviously the backbone of behavioral domains. Some of these are cognitive processes. Some of them might be considered sensory processes or internal processes, physiological processes. But it gives us into which to link our experiments and things that we can commonly search over. So using the activation likelihood estimation techniques, it's a quantitative coordinate-based voxel-wise meta-analysis method that is implemented in the BrainMap software, originally developed in 2002 and obviously extended tremendously. That's true, tremendously over the last couple of years, and applied to many, many different domains. We'll get to mental processes in just a minute. So anyway, in this analysis, we're actually looking at the coordinates as reported by a number of different papers and trying to identify what is the likelihood of an actual, common finding across papers. And we can, model these activations as a Gaussian probability, and then say, okay, across different papers, if we have these kinds of foci reported, and you can see there's quite a bit of variability here across different papers that are all doing the same thing. Very basic finger tapping kind of response. You would hope that you would see something basically in the hand area, but we actually get quite a bit across, quite a bit of the motor cortex. You say, well, where do we actually seeing evidence for in common across all of these different reported foci? By eyeballing it, you sort of go, okay, well, that's where most of them are, but can we do something a little bit more quantitative? So that's actually what they do, is they say, okay, let's take all of these foci and consider them as Gaussian and probability distributions, see where they overlap, and then do the analysis on that to say what's really significant across these different studies. And that's where you can actually identify with some robustness what we're actually seeing when we aggregate the literature. And of course the answer is always the brain is more complicated than we think, but it's a very nice system, and has been, you can use it to actually predict these different kinds of results and compare it to the actual results. Okay. And that's becoming more and more useful to identify these sort of consistent regions across studies. But of course you need coordinates. You need to actually combine your data across different tasks, or if not a specific tasks, for example, abductive reasoning. And abductive reasoning is classified under a larger constraint or a larger concept of reasoning and decision making. In itself, as far as I could tell, is not actually part of a tree, but I'm hoping that Russ will address that next week. But under reasoning and decision making, which we will appreciate as being a cognitive processes, he's got really quite a few cognitive processes that have been defined, so he gives it for each one. He gives a definition, and then what might be related to it, and then the actual brain regions that have been identified from experiments that seem to be annotated as being related to abductive reasoning. So if we look at reasoning as a larger concept, you can sort of see the different kinds of reasoning that he identifies. I don't have any other associations out here. But then he says, okay, reasoning as a cognitive process is measured by the contrasts within these tasks, these specific contrasts within these specific kinds of tasks. And of course, then you can go explore these tasks and identify what the conditions are and what the definitions are, and sort of represent for you what the brain areas are that show up as contrasts that are part of these reasoning results. And of course, this is the link between the task and the mental process. We have tried to, we've worked with him basically to try to make as many of these tasks as we can part of CogPo, and make sure that when we talk about conditions, we're actually working within that same framework, and are working with him to try to import this as much as possible. And of course, CogPo is somewhat agnostic about exactly what the relationship is between the cognitive task and the cognitive process. We sort of annotate the experiment with the behavioral domain, but don't get into the detail. Yeah, it's an interesting. So of course, the big goal, and Cognitive Atlas is trying to go this direction, Neurosynth is trying to go this direction, BrainMap is trying to go this direction. Well, at least, you know, I want to go this direction. So given current knowledge and available data that's relevant to, for example, brain circuitry underlying schizophrenic dysfunction, which is where I'm really interested, what is the next experiment to do? Where are the holes in what we know about brain function, and what are the experimental designs that we should be doing to address this hypothesis? Could we automate the process of generating a hypothesis and then determining whether the imaging data already exists in the publicly available repositories that would allow us to test this hypothesis? Now, we're getting closer to this all the time, but we're still a long way away. Many of you are probably familiar with this robot scientist, Adam, that came out a couple years ago, not working in neuroimaging, working in yeast genetics. And what they've done in this case is really build up a formal representation of what is known about a particular subdomain of genetics, and then build a robot that can actually generate hypotheses given the knowledge base of what is known about this, generate hypotheses, prioritize their hypotheses, design the experiment to test that hypothesis, run the experiment, it is actually a robot, run the experiment, do the analyses, turn those analyses back into knowledge statements and put the knowledge statements back into the knowledge database, and then go off and generate another hypothesis. It's unbelievable. I mean, it's a small domain of genetics, but it's really fascinating. They've been able to do that. That they've really achieved this idea of end-to-end automated experimental research. But why does it work? Well, they had a very, very well-defined subdomain in which they had a very constrained set of experimental designs. Unlike cognitive neuroscience, where if you can think about it, you can probably figure out an experimental design nobody else has done before. They actually had a highly constrained set of what you might do, and links, very, very carefully thought out links between what they were trying to work on, which is metabolic effects with metabolites and enzymes, and they had a very constrained set of relationships so that they could use abduction to actually infer what they're interested in, which is the hypotheses, the missing arcs between the enzymes and the metabolites and the genetics underlying that, so that they could represent the relationships between the experimental designs and the results very cleanly. In their case, find a path in this graph from the cell inputs the cell outputs, then the cell can grow. And so what they do is say, okay, if the cell grows, we can go back and actually reason about the metabolites and the other things that we're actually interested in. And then the results could be interpreted automatically as new knowledge statements with clear steps then to identifying the next experiment and identifying what's going on with the missing arc, what other arcs are still missing. We just don't that for cognitive neuroscience, but that's part of what we're all trying to do, in a sense. So, obviously we still need to harmonize the different cognitive process taxonomies. Gwen's been identifying, the things that are relevant for her issues. Janet's been identifying the things that are relevant for hers. Russ for Cognitive Atlas, us for the BrainMap and CogPo. So obviously we're all trying to interact with to harmonize that. And then, of course, the relationship between the cognitive paradigms, the results of those experiments, and the cognitive processes. Within BrainMap, we really sort of say, it's implicit. It's not really explicit what that relationship is. In Cog Atlas, they make it explicit as measured by, and maybe Russ can say more about that next week, but there may be other ways of actually modeling what the relationship is between the cognitive paradigm, or the contrast in the cognitive paradigm, or the analysis done on that contrast, and what do we actually out about the cognitive process as a result of that. And I think that's the thing I wanted to talk about. So this is just what we've been funded by and how we've managed to make. I'm going to go back and look at the chat screen. If you guys want to talk, we can. Now would be a good time. All right, I can see you. Should I mute my microphone or something so other people can talk?
No, I think that's great. Thank you so much, Jess. I had an issue unmuting myself.
Yes, I can hear you just fine.
Okay, perfect. So again, thank you, Jess. That's great, and thanks for filling in for Angie. Angie,
pinch hitting, Angie.
Yes. For absolutely doing it. Okay, so no, I think you did a fabulous. I think this is a very good representation. One of the things that I was wondering, and I think we should possibly get to some of these questions that maybe Angie didn't fully address in the chat during your talk, but one of the things that we keep getting hits on is the translation between that MNI space and Talarac space.
Really?
And yeah, because there aren't really, I think, really good answers to that on top of Google. So I think we need to just create a web page somewhere that says this is how you translate MNI space to Talarac space, and if that
there's standard stuff for that. It's not there's both linear and nonlinear translations.
Yep, exactly.
Actually, I can send you. Angie can actually, if you send the link of what you've incorporated into BrainMap, would be probably the best way to do that.
Yeah, I think that would be great. So that's something that we've gotten several questions on just in this. And we tried to point people your way, but that's. But the other thing that I'd like to speak about is this: the fact that this area of science is very hard, and what you can see when you're sitting over in the NIPS perspective, basically, is you can see that there are multiple databases, such as, we just did an analysis of SumsDB versus Braid. And out of the, thousands of papers that are in both of those, we found about 227 that essentially, those 227 essentially were in common. And we looked at those, and the way that they were described, and then the actual coordinates that were grabbed from the paper. So this is, all coming from the same papers. Those coordinates were actually a little bit different because SUMS does a transformation of those coordinates to a more standard space. And so again, I think these are important things, and these are things, you shouldn't take for granted looking at this data, because, it's wonderful to have these databases because this stuff is even exposed where you can get to it. Yeah, Braid and SumsDB are part of the NIF Data Federation, and we, literally looked at just the PubMed IDs that were in common, and we said, well, okay, let's look at this one. What did they say that the paper studied, and what did they say? And they're kind of related, but they're not the same. They're totally different.
Oh, that would be interesting.
That will be part of Ian's upcoming book chapter, but it's not yet published. So, so that is, that's really the gist, as you're saying. Trying to figure out what exactly is the heterogeneity and where might it be coming from. Because if these things aren't so similar in how we talk about them, then that really tells you that, there's going to be so much variability in the
well, and actually, that would be interesting from the point of view of the Braid and SumsDB. I would be very interested in seeing what papers they were and what the metadata is that SumsDB and the Braid reported about them. I mean, in the sense of if it's literally just a question of, okay, the points were, they used one algorithm and somebody else used another algorithm for transferring the coordinates into a new space, that's interesting but not so interesting. But if, one of them stored it under executive function and someone else stored it under attention, then I'd be really interested in understanding more about that.
Yeah, I think that's a really good.
Because the algorithmic differences in translation between these different atlases, they'll slide things around the brain by a centimeter or two, but they're not going to move it from the temporal lobe to the frontal lobe. I mean, unless they're doing something really horribly wrong. So, those are going to lead to moderate differences in the results. But when you start saying, okay, when we talk about inhibition, and now we're using one paradigm, whereas somebody else is saying, and somebody else says, no, we're talking about executive function, and so we don't even look at the same papers, or we're putting, we're using the same paper to make an argument about two different cognitive processes. Okay, now we need to actually clarify the situation, I think.
Right, and then we also move, again, from this kind of highly curated set to the neurosynth, which you brought up. And really, that's just a tag of terms that that they're using. So, how do you pull out those particular functions most effectively?
Exactly. You know what the writer of the paper thought the paper was about because they've used this word over and over again. But you don't necessarily know, unlike what Angie's done with BrainMap, you know that there is a point in BrainMap, you know that there is a point in the brain and it results from the comparison between this condition and that condition. So directionality even. Whereas with just bag of words approaches, you know that this point has to do with memory, but you don't know what the contrast was. Is it going up? Is it going down? And how does it vary? But that's actually something we've been trying to do here is to do sort of the NLP approach to say, can we identify what what a paper is about, and start to identify maybe some of the how do we map an abstract to an ontological term. So we could actually say, not only is this paper not just doing the bag of words approach, but actually saying, hey, this paper is probably about a Sternberg paradigm, or this paper uses a visual stimulus, so we can help narrow things down like that. Because I think that's got to be really nice. It's got to be where we go with this.
Exactly. So if we could map, these bags of words, at least these bags of words that actually match to a particular paradigm, then we could actually take the vast amount of data in neurosynth and start to actually do some of this potential reasoning. Now,
that's the theory.
I know we have issues with NLP, but you can't argue
if
with
it works, you don't knock it.
80,000 people.
Yeah, I mean, if it actually works, that's really good. I mean, at the moment, the drawback is we just don't have the training set that we need. Yeah, exactly. There's not a lot of overlap between the papers in neurosynth and the papers in BrainMap, and so we don't necessarily know what the correct answer is unless somebody actually sits down and expertly annotates those papers. I mean, for example, a lot of the text mining work starts with the PubMed abstracts, because those have all been tagged with the NLM mesh taggings by experts, and so what they're trying to do is actually tag it correctly.
Right. So one thing, just though, I mean, actually, we have a really nice webinar from Neurosynth, and so we spoke with him quite and extensively on this. And actually, the curator for SumsDB was the person who provided the human data set as a standard for neuroscience development. Good. So I do know that there was a very, very extensive training set. So he basically used SumsDB and then additional data on top of it, all coming from Aaron, basically. So a very interesting data set, and I would love to see how it matches up with any of the things that are in actual in BrainMap, because that's something that I don't know, because obviously I don't have access to that.
Yeah, neither do I. So, and neither does Angie. So it'd be interested to to get that.
Yeah, and see where the real differences are. But I mean, I can certainly pull down neurosynth data if you want it, so that we can start looking at where the bag of words and where the cognitive terms actually are starting to intersect.
Right. I mean, we've been trying to do that with some of the BrainMap repository data sets, but we're dealing with 350 abstracts within a particular behavioral domain and that kind of thing. So it's slow going.
Mm-hmm.
But,
so Gwen is asking, what are the mental processes?
What are mental processes? Oh, come on. This is talk number 3. We haven't figured that out yet. Well, Gwen, I thought you had sort of addressed that in your talk. I mean, they're the internal, the internal process, their internal process, and we're basically a label that we stick on something that seems to have a physiological output, but we are they're more useful. They're more useful and convenient than platonically realistic.
Right. Okay. Very good. Are there other questions?
Well, now next week we're going to be dealing with, we're going to have Russ's presentation.
Right, exactly.
Are we actually gearing up? Yeah, Beth, I will definitely put the slides somewhere. I'm not sure where yet. I think Angie has a location. But no, I guess my question is, are we gearing up in this series to get to the point where we can talk about mental processes and how they fit and how we would represent their connection? I mean, could we actually nail down a neuroimaging experiment hypothesis to the interpretation of results?
Yeah. I think that would be really the ideal and the goal of this thing. And I'm not sure that we'll get there in this particular series, but this is certainly the goal, and it's very important to state the goal.
Sounds good.
So, all right. Well, thank you so much. Thank you, everyone, for participating. Thank you.
Well, except that Angie actually put all those slides together and has done all this AL work, which is just really fascinating. So my apologies that we couldn't get the technology together, but nonetheless.
Yeah, nonetheless. Okay, so with Angie, thank you so much. And Beth and everyone, thank you. And we will come back next week to deal with
And I
Russ.
think, Beth, the answer is probably yes. But we can talk more about that, I think, later. All right.
I agree, Beth. The idea is how to cross-cut data. Okay.
All right. Ciao, guys.
Thank you so much. Bye-bye.
Bye-bye.